So I think about 4 or 5 weeks ago, we talked a little bit about Designer, and now we're really happy to announce that we actually released a new version of Designer, which will help people get started with designing a custom single-cell DNA panel design. So this software really is simple to use, and what you can do is you can begin to ask biological questions which are relevant to your cancer research, or if you have a CRISPR model system. It allows you to completely customize the targets that you want to go after. So with that, I just want to back up a little bit and provide a little bit of an explanation or a background about what exactly single cell DNA sequencing is and why it is the way it is. So if we take a step back and we think about bulk sequencing, you really have to have the best fit for the application. So there's sort of 3 flavors of bulk sequencing. You can have really targeted sequencing panels, which are typically amplicon based, which go after very specific regions of the genome. You have exome sequencing, which will just look at the exon regions of a particular gene, or you can have whole genome sequencing, which is trying to cover every base in the entire genome as best as possible. So this is an example from the University of Washington, which just kind of highlights the differences between those. So if we look at a particular gene or DNA region, you can see you have exons and introns, and if you do whole genome sequencing, you're trying to cover every part of that gene. And the intron regions may not be particularly interesting or useful to your research, and then you're just wasting sequencing reads on those. And so they've created quite a useful example is if you look at the whole genome, which is illustrated as this apple, only about 1%, and that estimation varies, but it's a small percentage of the whole genome, which is the exome, which is protein coding regions of DNA. And so it's not particularly useful in many applications to understand what's going on in the whole genome. You really just want to understand what's going on in the whole exome. If there's copy number changes or single nucleotide variants, which would encode for a protein change, which has a functional effect on that cell. It's also important to note that even though that's about 1% or a few percent of the genome, it's still about 30 million bases. So it's a pretty heavy sequencing requirements to think about if you ever wanted to translate this to single cell. So again, so this is doing targeted versus whole genome sequencing. You really want the best fit for the application. So if you look at whole genome sequencing, you're going for 100% of the genome. If you're going for whole exome sequencing, you're going for a few percent of the genome, just covering those exons in a gene and ignoring the intron regions. It's important to note that if you do whole exome sequencing, there's usually some sort of bait and capture system that captures those regions, and that has its own technical challenges to do. And then finally, you go to amplicon-based sequencing, which is really looking at a very focused, targeted regions of the genome. And that's exactly what Tapestri is. It's looking at very small targeted regions of the genome, which are biologically relevant to a particular type of cancer or a particular disease of interest. And asking specific questions about what's going on in these particular sets of genomic regions. And then the reason that Tapestri uses targeted sequencing is that it's the best fit for single cell. As I mentioned, if you're even just looking at a few percent of the whole exome, that has very high sequencing requirements. And what Tapestri is an amplicon based, it's just looking at genes of interest to make it practical to sequence across thousands and thousands of cells. And so that's why Tapestri and Tapestri Designer software helps you build up targeted sequencing panels just for the regions of interest and alleviates a lot of the sequencing requirements to make it economically practical from a cost benefit perspective to actually sequence thousands of cells. And so how does Tapestri really work, and why are we the only DNA sequencing platform? Is that we have a unique 2-step workflow. And this is just a schematic of what's going on inside the microfluidics of Tapestri. You input your cell sample into that, and each cell then becomes encapsulated into a droplet. And in the first step of our process. That cell, which is now inside a microfluidic droplet, is lysed by a protease cocktail, which is proprietary to Mission Bio, and then inactivated. And then from that point on, everything within inside the cell is now accessible to our chemistry. So the DNA, as you can imagine, is coiled and packed into chromatin. It really requires that protease step to free up that genomic DNA so it can be accessible to our chemistry. So that cell lysate in the second step goes into a second droplet in which we introduce now a bar-coded bead and then our reagent mix, which is highly optimized for high plex targeted amplification. And so you can see in that second droplet, now we can amplify up just the genomic regions of interest, leaving behind the things we're not really interested in. I just wanted to also highlight here is that the chemistry of Tapestri is very well suited to do this targeted amplification. This is an example of 3 of our catalog panels. And you can see on the x-axis, this is the increasing reads per cell per amplicon. As those increase, you can see that on the y-axis, we have the number of amplicons that have at least minimally 10 reads. And you can see we have a very high coverage rate on our catalog panels. And then that feeds into our software. And our software's, we have Tapestri Designer, of course, to build panels. We have Pipeline and we have Insights. And of course, we're going to focus on Tapestri Designer today. But I just wanted to highlight that we have a complete end-to-end software solution for our panels. And our Tapestri Designer is very simple to use. It's intuitive. It's and I'll highlight that. I'll show you an example that you can just create a panel within minutes that answers very specific biological questions. It's very high quality. So we build custom panels that are really specific exactly to our chemistry. And we've done that through a bunch of machine learning and powered by AI. So essentially, every time we run a panel and we gain more information about how amplicons behave in our system, we feed that information back into Designer and it gets improved with every design. And that's absolutely true with the launch of Designer 2.0, is that we've made and optimized our design engine to be smarter each time that we are building up custom panels. We can also customize existing catalog panels in our system. So you're not just starting from scratch every time. So if you guys have interest in one of our catalog panels, but you really want to introduce additional targets or remove content, you can do that in Designer, and I'll show you how to do that as well. And then what we're really talking about today is that we've now expanded to whole genome coverage. So the initial 1.0 version of Tapestri Designer only really covered the exome, and now we're introducing whole genome coverage for both human and mouse genomes, recognizing that some of our customers want to translate some of their human research into animal models. We can now support that with our Tapestri Designer. And as always, if you have any questions, we're more than happy to have technical discussions anytime. And so you can always reach out to us, and we're more than happy to answer any questions about what kind of panels you can build or kind of applications that can be supported on Tapestri. So actually getting started with Tapestri Designer is very easy. You just go to our website, you set up an account in a couple minutes, you can enter your targets, and I will show you how to do this either through a CSV or Excel file, or you can just copy paste right into Designer. And then you just hit submit, and all of that will be pushed to our Designer, and then it will contact you back with an email within a few minutes or a few hours, depending on what kind of design and the size of the panel that you're trying to build. So a couple of slides ago, I showed you some performance data from our catalog panels. This is just also highlighting the first 3 sets here are the design coverage, meaning that from the targets we submitted, how much of that can I actually capture in a custom panel design. And then in the dark blue bar, we're showing you actually the wet lab performance on uniformity. So this is just showing you that whether it's a catalog panel or a custom panel, our design engine does quite a good job of getting the best uniformity, which translates to sequencing efficiency. meaning so the higher the uniformity means that my best performing amplicon and the least performing amplicon, there's not a huge disparity between the two of them, and they give you quite good efficiency of sequencing, as well as covering all of the variants of interest. So just want to spend a few minutes talking about Tapestri Designer 2.0 and what's actually happening when we've moved to this upgrade. So as I highlighted before, we're moving to whole exomes support. So we previously were only able to cover the exons of genes, and now we're moving to whole genome support. And so what does this actually offer to you, the customer? You now can go after things like non-coding RNAs, which potentially are interesting to particular cancers. And so non-coding RNAs are genes which express an RNA transcript, but are never translated into a functional protein. And so many of these RNAs actually have biological relevance, particularly in cancers. So now you can enter those regions. You can also go after things like gene promoters, which are outside of a coding region that may be of interest. If, let's say there's a mutation that occurs under a transcription factor binding site, you can now capture that in a custom panel. And then as well as you can do on-target and off-target site analysis for CRISPR gene editing. So we have a number of customers who are interested in looking at if they're making a particular gene edit at a locus. Now you can see that, yes, I am making that particular gene edit, but am I making any other edits at off-target sites? And those off-target sites typically will sit somewhere outside of the gene coding region. As I showed you in the first slide, 99% of the genome, or roughly thereof, is not coding for any protein. So this is an important change, particularly for the CRISPR gene editing customers. And now also we have genome support for mouse. The coordinates that are supported now are MM10, which is the latest version of the mouse genome. And this is important for translational models of disease, or if you're working in some sort of cell model or organoid system. This really enables kind of more of a discovery mode where you can have your particular mouse system and model of disease, and then create a custom panel just suited around that particular mouse model. And I think in the coming weeks and months, you'll see a number of publications coming out which highlight the utility of this from some of our customers. And then finally, as I mentioned, again, we have a really optimized design algorithm, which is really suited just for our chemistry. And with the update to Designer 2.0, I think you'll notice improved target coverage. So we've kind of reworked the design rules, which enable a much higher coverage than previously was possible with our design, because we have now a little bit more flexibility of where we position the primers within that panel. And so you can get much better coverage than previously were able to. So in my last webinar, I did show this slide, but I just want to kind of refresh everybody if you hadn't had a chance to attend. There's 3 types of inputs that you need to provide with any amplicon design. You need the type of input, the identifier, meaning whatever the type of input you have to give it what that piece of information is, and then you can give it a unique name, which is an optional. So these are the types of inputs which are supported in human. So you can input an official gene ID, and there's some examples of GAPDH or SOD1. You need a transcript ID, which is from RefSeq, and the source there is NCBI as well. You can also put an HG19 region, so you can designate any chromosome in the genome, and then the base positions in HG19. And that's particularly useful if you're looking for a particular hotspot that you want to cover, or a small region or indel. You just provide the coordinates of that, and then the designer understands that you're trying to target that region, and we'll build an amplicon around that. It also supports common databases, so things like DB SNP IDs, cosmic variant IDs, or HGVS IDs. It can put those in. Important to note with HGVS IDs, you can see this one I've shown as a genomic example, but if you're putting in a protein-based change, an amino acid change, just be aware that based on the different model of the transcript of that gene, there could be multiple sites that are relevant. So just be aware for HDVS, be sure that you're targeting the right genomic coordinates based on the different transcripts, because there could be several different transcripts from one gene targeting to that one locus. Here I'm just highlighting where you can find information from different genes, panel content. I mentioned DB-SNP. You can go into cosmic databases, look for your particular type of cancer, and then utilize that to identify the mutations that you have an interest. We've also built a number of pre-designed panels off the Cancer Genome Atlas. That's another great resources where you can identify several hotspots of interest. In addition to these public databases, you can also always use bulk sequencing data. So we have a number of customers that will first perform bulk sequencing, identify the variants of interest or novel variants that they want to investigate, and then utilize that to build out a custom panel. And then certainly there's also publications and other things like that where you can sort of build up a list of targets of interest. Real quick, I'll just cover a couple of tricks and tips. Like I mentioned, the HDVS IDs, making sure that we're targeting the right genomic spot. It's important to note that our chemistry, we can't tile completely. You can't overlap two primer sites. That will create kind of a hybrid amplicon, which really is not ideal. You don't want to have that. So all of our primer sets are spaced by one base. So you never get 100% coverage of an entire gene. Now, if you're just going after exons, that's not really an issue, because typically you can create primers that will cover all the exons. And I'll show you an example of this in a couple of minutes. It's also important to note that with single cell DNA sequencing, we're starting with just 2 copies of template, right? There's 2 copies of each site in the genome. And so some considerations apply to PCR primer limitations, such as %GC, repetitive sequence. And so our designer accounts for this in the stringency of design. So if you're putting in a gene and it happens to have very high GC content, you'll say, why am I getting such low coverage? It's really because we've optimized our designer to really have the best design rules that we think that it will operate within our chemistry. Now, if you must have coverage, you can always have a discussion with us and we can attempt to rescue back some of that coverage. But really our designer is set up to have the best outcome, meaning that we'll limit the design to the regions we think will operate best. Next, to identify copy number changes or loss of heterozygosity events, you're going to want to put between 5 and 12 amplicons within that region of interest. And then also place a similar number of amplicons flanking those regions. So it's essentially when you have flanking amplicons, that's like the control region. So if you wanted to call a copy number change, you'll have your region, which is diploid, and then you'll have the target region of interest where you expect the CNV change, and then you can call that based on reduction in reads mapping to those regions. And I'll show you an example of that when we go to the live demo. And then finally, carefully review for critical mutations in the panel design. I'll show you that you can do this quite easily in Designer. Just by clicking a button, it will take you to a visualization of those amplicon regions. And it's very important to get granular with the content details for your panel. The more specific you are with the inputs, it really helps reduce the coverage to just the important targets. So you're not just creating amplicons and that aren't particularly useful to you. And that helps you save dollars when you go down to sequencing those genomes. And finally, as always, you can always reach out to support. We're happy to talk through any of these points and give you additional tips and tricks on how to design. So with that, let's go to a real example. So this is from a publication looking at acute myeloid leukemia. And it was actually showing that there's particular deletions or amplification events which are expected within this genome. And for here, I'm really going to show you an example of a deletion in 5q. So you can see in the P arm, there's not really a high degree of copy number change. But if you look at the Q arm, sorry, of this, there's quite often a deletion event that happens. I'm going to show you real quick how we can build up a custom panel which would address that question. So first step is really decide what you investigate. So I just gave you an example. So in this case, we want to look at a couple of SNVs. So I'm interested in P53, very commonly mutated in cancers. And then an AML DNMT3A is also a frequently mutated gene. So in this case, my custom panel, I'm asking, are there any mutations in P53 and DNMT3A? And then also I want to understand, are there copy number changes also going on? So we just saw in that publication, the 5q deletion happens quite frequently in AML. And so I'm going to ask, can I look at a couple of genes which are in the 5q region and the 5p region? And then I'm going to pick another gene that's completely outside of both of those regions. And so here's my list I've built up on the right side of the slide there. So my inputs are now saying, these are all genes. So I can say, here's a gene. The official identifier is TP53, and I've just called it P53 as the name. And I've done the same for DNMT3A. I said it's a gene, cover as many regions in DNMT3A as possible, and then also just call that DNMT3A. Now, to address the copy number change, I've put in a gene. This NRG2 gene is actually on the 5q arm. So if there was a deletion event, I would detect that because I would have reduced number of reads going to my 5q arm. I've also picked a gene RAD1, which controls basic excision repair on the P arm of that same chromosome. And then finally, I've picked another gene, which is completely outside of that chromosome as kind of another control. And that's totally optional. And there's many different ways we could configure this and we get really specific. Or we could even, if we wanted to do the CNV event, we could just pick regions that are equally spaced across that arm and then just put in those regions in my design. So with that, I'm going to step out of this presentation real quickly and I'll go over to our designer software so you can see how that gets put through. So if you go to tapestrydesigner.com, you create an account, you log in. This is the home page, which you'll see. And you have two options. You can target with precision. So I can click here and I can target with precision. I can see my catalog panels. I can see a number of other pre-designed panels and the metrics around that. And what you can do is even go in here and click one of these. Say if I go into our AML panel, I could hit the copy button here. I could have all the content in our catalog panel, and then I could edit, I could copy it, and then go into my custom panel design and then start to edit that panel by adding or deleting amplicons from that panel. In this example, I'll go ahead and just create a completely custom panel. So I'll go back to our home page and I'll hit Launch Designer. And from there, I can hit Create Custom Panel. And this will bring you to this page. And you can see the assay type is currently DNA. It's going to ask me which genome am I looking at? Am I looking at human HD19 or am I interested in mouse MM10? This is going to be a human panel design, so I'll hit HG19. It's now selected that. You can see our amplicon length range is typically 175 to 275 base pairs. And then the only other input I really need to offer it is what am I trying to design? So I can either upload a file with those three inputs, remember the type, the identifier, and the name for those, or I can paste those in. And so in this case, I will go back to my webinar slide deck. I will go easily highlight those regions. I can copy them and put them right into Designer and then hit Submit. And then it'll ask you to name it. We'll just call it Custom Panel. You hit Next. And then it'll say that it's processing. So when it says it's processing, what's happening in the back end is that the designer is now looking at my list of targets and trying to match these up and validate that those are true targets. So it's going to say anything that I've put in as an identifier, is that a real gene ID? If it's not, it may be a typo. Maybe you can just edit and clarify what is the actual target region. And so if it just takes a few moments and I've hit refresh, and now it says, okay, good. 5 of 5 of my targets are successfully added. I can see all of those now are in there. And then it has the names that I've given it and the actual IDs. So you'd hit continue and then submit, and it would go ahead and design. And so that takes a few minutes. I've actually already gone ahead and done this. So I'm gonna go ahead and show you the results here. So now I can see I have my gene, the name I've given it, and the identifier. And you can see it has a little arrow here. It's just warning me there's potentially a few high GC amplicons. It's just to be aware that some of the regions in that gene may be a little bit high GC. Typically they work, it's not an issue. We just like to be upfront and show you that may be regions are a little bit more difficult to do single cell DNA sequencing on. As I mentioned before, we can get really good coverage of whole genes. So you can click on this link to visualize the design. It will immediately drop you into UCSC Genome Browser. And you can see I have 3 custom tracks that are now incorporated on top of UCSC Genome Browser. You can see I have the different transcripts of DMT3A. In blue, it's showing me these are the regions I submitted for design. So even though I just called out the gene, Designer will know that we're going after the exons in that region. So it will submit those for me. In green, it shows me all the regions that I actually am covering. And then in red is if it wasn't able to completely cover an exon, it will highlight that in red. So just be careful when you're looking at your design. Make sure that there's no critical mutations which are lying in those red regions. If there are, you can contact us in tech support and we can start to maybe help complete those designs. So you can see here that I've shown in this particular exon, you can see we submitted the whole exon in this region. It covers almost all of it, but there's a little sliver of red here, right at the end of that exon, which is not covered on that design. So that's the kind of granularity. And as I mentioned before, it's the HGVS IDs. If you really want to be sure, you can zoom way in UCSC and you can go all the way down to the amino acids in that region. So if I keep zooming, maybe I have to hit 3x here. Eventually now I can see the amino acids here. And if I go down a little bit deeper, I can see my covered regions, and it will actually have the base positions of those amino acids under each of those transcript IDs. So you can see now, zoom in so everybody can see. You can see based on different transcripts, now I have a leucine at site 405, but it's actually called 594 in another transcript model. So just be sure that you're covering the right ones, or you design amplicons to cover both sites at L405, so you can capture any mutations that may result in a change in the protein of that gene. Okay, so finally I will jump back to my presentation here. So that's that fast. Within a couple minutes, you can create a custom design, and you can do many different iterations of the design to get it to the point where you really want it to be the panel that you want for your single cell sequencing. Finally, just wanted to highlight, once you have that design all completed in Designer, you can also output this as a whole design package, which you can bring into other software. So it will give you bed files and tab files, cover it with all the information about the panel, whether it's the chromosome positions, the insert sequences, all of that can be output from our software. So with that, I will wrap it up. So happy to take any questions about design process, but just how to get started or next steps is you can just easily go to tapestrydesigner.com. Account setup takes just a couple minutes. I do want to highlight that everybody who's kind enough to come and attend this webinar will be receiving a special promotion discount on custom panels should you decide to try out a Tapestry. I'd also ask that if you're interested, please follow us on social media for all the news and events. We're doing a whole bunch of these webinars and different events online. And if you have any questions, you can either reach out to our FAS team at support@missionbio, or you can email me directly at flynn@missionbio. I'm happy to answer any questions offline if people have them later on. So with that, I'd like to thank you, and I'd open it up for any questions that people might have.
Great, Jim. Thank you so much for taking us through Tapestri Designer and all the new features in 2.0. I think our customers are going to be really excited to be able to do the whole genome and mouse. So we had a couple questions come in. Can you comment on how to design for CRISPR edits where you can have tremendous variation from cell to cell in the types of indels that are created?
Ah, that's great. Yeah, so Tapestri Designer, typically people are doing on target, off target. So they're looking at their BAM sequence and then padding it with several bases, just so the amplicon's positioned. You really want the amplicon created so that the edit site's towards the middle of the amplicon, favoring like one end, so that you get good coverage from the read one, read two. If you have or expect really large indels or rearrangements, we handle a lot of that in our white glove team. That's a little bit more complex design, and so it requires a much more manual approach. So if you do have a CRISPR panel, please reach out to us. We'd be happy to take that in our white glove team, and we have a lot of experience building those kind of indel-based panels.
Great. What is the biggest panel that you can design?
Ah, that's a great question. In the software, I believe it's about 1,000 amplicons. We currently sell panels up to 500, but we have done large panels. The real limitation there is not so much on the chemistry. It becomes an issue of sequencing cost. Gets up quite significantly. So if you can imagine, for every amplicon we create, we're targeting 80x coverage. So that's 80x times the number of amplicons in the panel times 5 to 10,000 cells, and extrapolate out the sequencing cost from that. But if you would like to build something that's quite large, we can certainly talk about that in our custom group.
Great. We had a question, so it's really cool that you could go into UCSC to look at your missed regions in more detail. But is there just a list that's provided that will tell you what's missed?
Yes, absolutely. I mentioned that you can download a design output file. It's essentially a zip file with all the regions. And within that, there is one of the files that has all the regions which are not covered in the design.
Okay, great. And then I think we have one final question. So if I have an SMV that's really, really important, if it's in a panel that's of 50 amplicon size or 100 or 500, will it always work?
The only risk there would be from primer-dimer interactions, and our software does have a module which looks for that in the design. So it's quite difficult to predict primer interactions, but I don't typically anticipate that there would be some primer-dimer issue with the panel. If there is something that's absolutely critical to design, and for whatever reason the designer is not covering that, you can always reach out to our tech support team. We can go in and try to look at that issue manually.
All right. Well, I think we've covered all the questions. So Jim, thank you again for taking us through Designer, and thank you for all of our attendees for joining us today.